Good afternoon. I'm Gary Morishima. I have the pleasure of introducing the fifth PSC seminar of a series on environmental change. The seminar is going to be about the International Year of the Salmon 2022 expedition, where scientists from Canada, Japan, and the Pacific Republic of Korea, and the Russian Federation, and the United States work together to try to understand the ecology of Pacific salmon in the North Pacific Ocean. Before we get started here, I just wanted to inform the PSC family of the passing of a real salmon warrior who was very concerned about environmental change and the future of salmon. Terry passed away yesterday. He was a longtime member of the Southern Panel. And knowing Terry, he'd basically say there's lots of work, lots of hard work we've got to do, so let's just get on with it. The 2022 Pan-Pacific Winter Expedition had the purpose of improving understanding of how environmental change in the North Pacific Ocean affects abundance, distribution, migration, growth, fitness, and survival of Pacific salmon. And as I mentioned before, this is a cooperative effort by scientists from 5 member countries of the North Pacific Anadromous Fish Commission. We have an outstanding group of 4 prominent scientists who are going to share their perspectives and their experience regarding this. We're going to have a tag team presentation today. So I'm going to briefly introduce all of the presenters. The first is Dick Beamish, who is Emeritus Scientist of the Pacific Biological Station in Nanaimo. He was the director of the PBS from 1980 to 1993. He's received numerous awards and recognition for his contributions. Dick was very heavily involved in all aspects of the high seas salmon surveys that were conducted in 2019, 20, and 22. Another one of our presenters is Brian Riddell, who is the science advisor for the Pacific Salmon Foundation. He was the past president of the Pacific Salmon Foundation and worked on the Salmon Sea Survival Project. Like Dick, Brian has received numerous awards for his work. I first met Brian, I think, in the early 1980s when we were still in the process of negotiating the Pacific Salmon Treaty. But Brian's been very instrumental in working with Dick in organizing research for the Gulf of Alaska surveys, and in particular trying to evaluate the impact of using gill nets to compare trawl nets during the 2022 series. Brian also co-authored the UN Decade of Ocean Science proposal to continue this effort. Our third presenter is Laurie Weitkamp from the National Oceanographic and Atmospheric Administration in the United States, and also with the Northwest Fisheries Science Center. She's been involved in scientific basis for West Coast salmon, is a member of the Coho Technical Committee, and her research has focused on the ecology of salmon in estuarine and marine environments. She participated in the 2019 Gulf of Alaska expedition and also in the 2022 High Seas Survey. Our last presenter is Chrys Neville from the research. She's a research biologist with the Department of Fisheries and Oceans with over 25 years of experience in studying the marine ecology of Pacific salmon. She participated in the 2019 Gulf of Alaska expedition and helped organize the 2020 expedition. She was also involved in the 2022 survey. Her main focus has been on comparing growth and conditions of salmon caught during the winter expeditions and doing a comparison of that with the juveniles that her program has been collecting in the coastal waters of British Columbia. So with that, I'm going to turn it over to the tag team members.
As Gary mentioned, we are talking today about the International Year of the Salmon 2022 High Seas Survey. And what we're really planning on doing today is talking about the motivation, the methods, and then some initial results. And you will see, I think becomes obvious that a lot of the data is still, we're working on it. Samples haven't been processed yet. In fact, we're still trying to get them to the right people to process them. So these really are very initial results. Next. And we just wanted, for those of you who aren't familiar with it, this was really the vision for the Pan-Pacific Survey. We were hoping to have a whole series of boats stretching across the entire North Pacific. And what ended up being the actual survey, we had 5 ships, didn't do quite as much, but it ended up being a well-publicized international multi-ship survey of high seas Pacific salmon habitats across the North Pacific Ocean that we conducted this past winter. And it was really, I don't think this has happened for a long time, that large an effort with that many ships. So Dick, take it away.
So what you've heard is the purpose of the 2022 International Year of the Salmon, but this is a little bit different because the original concept of the International Year of the Salmon was in a report that Brian Riddell and I, along with a whole bunch of other people, put together for the North Pacific Anadromous Fish Commission in 2009. And then we wrote a couple papers about the importance of it, and a little bit different from the purposes that you've already been told, but similar in the sense that number one was the original intention of the International Year of Salmon to have international teams of, let's call them researchers, work together to improve the understanding of the winter ecology of Pacific salmon. And so the concept was to work together as international teams. But what happened was, as the IYS was being organized, Brian Riddell and I organized our separate expeditions. And these were privately funded and privately organized expeditions, as you've already heard, 2019, 2020. And then we organized a third one for 2022. But those 3 expeditions were all privately funded and privately organized. The intent of those 3 expeditions essentially were to see if winter surveys could improve the forecasts of Pacific salmon returns, and specifically to British Columbia. Now, I don't know how many people know that Russian scientists, led by Dr. Shuntov in the 1980s, had a similar program in which they were looking at using fall surveys to forecast the returns of pink and chum. And it took a while, but they were successful. And for many years, decades or 2, they were very good at forecasting returns as a result of these surveys. So the second objective we had was, could we have these winter surveys, and would they improve the forecasts? And then you'll see in the next slide, which is quite amazing, that there is a third reason for them. And the third reason is why, at the end of this presentation, our presentations, we'll talk more about the need to continue them. So if you go to the next slide, I don't know how many of you have seen this, but there really has been a total collapse of the commercial salmon fishery in British Columbia. And these are the percent of Canadian, all species of salmon caught in the Canadian commercial fishery. And in the last few years, the percents have been disastrous, really. In 2021, British Columbia commercial fishery represented only 0.2% of all the salmon caught in all the commercial fisheries in the North Pacific. So we'll talk a little bit more about that later, but I will suggest that if we if we think that we have more or less all the information that we need in terms of managing salmon in British Columbia, this represents a substantial mismanagement problem. But if we accept that what this really shows us is that there is a lot more to learn and we need to see a bigger picture. We'll talk about that later. And I think you will see in the following slides that we have made major discoveries in the few years that we've been doing that. I'm going to talk about a couple of them. So the next slide. That's Brian Riddell's slide. Where is Brian?
Brian, you're muted.
No,
There you go.
I got it. Okay, so the third vessel that, or the third expedition and the fifth vessel that we're referring to, Dick and I intentionally chartered a commercial vessel and rigged it for gill nets and long lines. The 2019 and 2020 trawl surveys were really trial and error. They were really experimental because Canada had not used trawls in the surface water in the open Gulf of Alaska. So it was very much a matter of let's try it and see. We added the gill net and long line because, to be honest, the catches that we had were really surprisingly poor. I mean, low in our opinions, right? So the trick then is, well, how do you evaluate that in an open environment like that using a new gear? And so how can we test that we're actually getting is truly representative? Dick and I were fully aware of the past high seas research by the Japanese using their surface research gill nets. A couple of other things that were really interesting in the early period is that the catches were very different between day and night use of trawls. And our impression is that the species were very heterogeneous in their distribution. They didn't, you'll see that they didn't overlap very much at all. And then the final point is that we have had people calculate the total abundance based on our surveys, but they apply a catchability to the trawl that is really based on the Russian experience. The trick then for us would be, could we apply an alternative sampling gear to try and get a sense of whether that catchability is appropriate in that environment? So there are a number of reasons for us trying what we thought was a proven technology and one that is an alternative approach. So we actually went and got Japanese research gill nets. They were made by the same company in Japan. We were concerned about fishing in the winter with gill nets. We were advised that this is possibly not going to work. And if that was the case, we were very concerned that we could lose them. So we actually employed satellite buoys at both ends that would allow us to track every gill net that we fished. And the other thing with gill nets, and this came out in real spades because one of our major funders dropped our support, was the concern about past bycatch in gill nets. That is acknowledged to be an issue, but we saw minimal bycatch at all. And I attribute that a lot to the regulations under NPAFC. We only ever fished 2, but the maximum we could fish would be 3 nets, and every net cannot be over 2.5 kilometers in length. That's fractions of the amount of gill net that used to be put out by commercial fishing. And the only other change we made is that our fishermen, when they saw the nets and the strength of the ropes, they basically said, this isn't going to work. And so we modified the nets by adding very, very strong, basically what people call metal rope, but 11 millimeter Spectra rope from Europe to add very strong strength to the float line so that we didn't lose the nets when we hauled them in. So I'll leave it at that, Laurie.
Yeah, so I'm going to talk about methods and results next. So as I mentioned, we had 5 ships out there. This is showing the track of each of the ships, and they're color coded. So the Northwest Explorer is in pink, Tinrose in orange, Shimada's in green, etc. So we had 5 ships that sampled a total of 131 stations across 2.5 million square kilometers of the ocean. So this really impressive effort. On each of those ships, we did three things. We did physical oceanography, and this was really a full oceanographic workup looking at all kinds of things. And I just highlighted the environmental DNA. So those of you who aren't familiar with environmental DNA, or called eDNA, all organisms that are in the water slough off DNA all the time. And so you can take a water sample and filter it, and then look and see who left behind DNA and identify what it is. And I think, especially when conditions were really rough, like all of us saw it one time or another out on the ocean, this environmental DNA allows you to sample without having to put the net in the water. And it's not a true and tested technique. There's a lot of questions that we're learning, but it provides a lot of insight. We also did full biological oceanography. All of us used this vertical bongo net, a couple other nets, and then we all fished. So either we had a surface trawl, or the raw spirit used the Japanese style, research style gill nets for it. Next. And this is just a list of the types of measurements and samples that we collected from each salmon that we caught on board, assuming we had enough time to process it. Standard things like lengths, weights, scales, otoliths, cutter wire tags, stomach contents for diets. And then we actually used a lot of advanced technology. So genetic stock identification is a huge one. And that's something that I think is somewhat unique compared to the historical high seas work, in that we can tell exactly where each fish comes from based on their genetic stock identification. We're also doing things like taking gill tissue to look at the up and down regulation of genes, blood for insulin-like growth factor, which is an indicator of active growth, and then a variety of other things. And some of them, especially the bottom three here, or excuse me, the bottom two, eye lenses for elemental analyses, and opercular invertebrae for lifetime hormone levels and parasites. These are really experimental approaches. And so we were able to provide samples to look at this and say, is this work? Next. And of course, we didn't only catch salmon. We caught a lot of other stuff as well, primarily these myctophids or lanternfish, squid, jellyfish, other fishes. Everything was identified, counted, take lengths and weights. And then we also took a variety of tissues for things like diets, bioenergetics, microplastics, voucher specimens for genetics. You need baselines. And the fleet caught two salmon sharks, which were both tagged with archival tags and released. And I know the one that we caught on the Shimada swum at least 1,000 kilometers since we released it. So it looks like it's doing well. Thank you. Next one. So this is just some pictures showing processing on the Shimada, and I think it looked very similar on the other big ships, perhaps not so much on the smaller ones, but labs with lots of people working, trying to get all these samples, get the catch worked up. As far as the conditions, this is just showing the temperature on the left and chlorophyll A on the right. These are the means for February. And you can see that for the temperatures, these purpley colors are very cold, 2.5 degrees centigrade, all the way down to the bottom of the figure where we're getting 10, 11 degrees. So a really wide range of temperatures, north-south, perhaps not as much east-west. And I have the chlorophylls showing as well. And I honestly don't know what that means, except that the gray areas are when it was cloudy and they can't see what the chlorophyll is. This is all satellite derived. If you go to the next slide, it just compares what we saw, these temperature and chlorophyll compared to 19 and 20. And the top row are temperature anomalies. So these are degrees above or below normal. Again, these are monthly means for February. And you can see that in 2019, it was actually quite a bit warmer than the other years. This orangish colors are 1.5 to 2 degrees centigrade above average. In 2020, the temperatures were pretty average, not high or low. And then in 2022, we again had this relatively cold water. So temperatures 0.5 to 1 degree below average for that time of year. And you can see in the bottom row are just the chlorophyll A's for the 3 years. And to me, they look pretty similar. I'm sure oceanographers would disagree with me, but I don't think it was really exceptional as far as this primary productivity. between the 3 years. Next. So these are the catches. So each figure, each map is showing the catch of a species. So we've got chum, sockeye, chinook on the left column, pink, coho, and steelhead on the right column. And these are just catches by the trawls. This does not include the gill net. And where there are X's means that that ship fished, but they didn't catch any. And then the size of the figure indicates how many were caught. And you'll see for both chum and sockeye, there's some triangles there. Those are where they caught more than 50 fish in a single haul. And it's interesting that in general, there tended to be larger catches of chum and sockeye just south of the Aleutians. But if there were a lot of chum salmon, for example, then over further east in the Gulf of Alaska, there were also generally more chum salmon. Some of the things like chinook, we caught very few of those, 18, which is what we expected, given that chinook are the least abundant of the salmon in the North Pacific, and they tend to be pretty deep. So there were probably salmon we missed that were below our nets. What to me is really interesting as well is the steelhead catches, in that none of the trawls caught steelhead. We were trawling at the surface. You can see the floats at the surface, but there's a little bit of a gap between the surface of the water and the where the top of the net is, and all 57 steelhead were caught by the gill netter. And I think Chrys is going to talk a little bit about that more, and which really indicates that these are incredibly surface-oriented fish. We already knew that, but to me, it's just staggering to see how many think about how many we probably missed out there. Next. So this is a pie diagram showing the catch of all the salmon across the entire survey together. And you can see the dominance of sockeye salmon. Admittedly, there was one haul that I believe the Russians caught that was over 600. I think there were Ocean Age 0 sockeye, but still there were a lot of sockeye out there. And second most abundant were chum, and relatively low numbers of pink and coho. Mentioned the steelhead were only caught by the gill netter. Next slide. What's really interesting is if you compare that, the catch of salmon that we got versus what the commercial catches across the entire North Pacific. So on the left is the whole North Pacific catch, commercial catch. This is the average over the last 20 years. And you can see the clear dominance of pink salmon. And you compare that to what we caught, and it's like, where are the pink salmon? It's really staggering, the difference. And you might say, well, we weren't, we didn't sample very much over in the Western Pacific where we know there are a lot of pink salmon. So what if we just look at US and Canadian catches, which is the pie diagram on the right? And you can see again, there's still this huge pink salmon. So one of the big puzzles to me is, where are these pink salmon, especially given the thought that they may be interacting with other species out there. So this is now showing the catches. Each pie is the overall catch from each ship. As I mentioned, Tin Row got one huge catch of sockeye, but they got others as well. But all of them look more or less the same. The sockeye were everywhere. Everyone had this large dominance of sockeye. Perhaps the one exception is this raw spirit, the bottom right figure. That was the gill netter. And the reason it looks really different is that big green area, which is steelhead. So they're, again, the only ones who caught steelhead. And if you take that out of there, it looks a little different. It also seems like we didn't catch very many coho, but I believe that either Dick or Brian mentioned that a lot of the coho are caught at night. And it sounds like the Franklin in particular didn't catch very many, or didn't sample very much during the night. So that may affect catches as well. So there's some things we need to sort out in really making these comparisons. If you also look at across years, so this is showing, again, the pie diagrams of the composition and then the numbers of fish in 2019, 20, and 22. And I have the maps down below showing where those occurred. So 19 and 20 were both in the Gulf of Alaska, compared to our entire survey area, which is much broader than that. Again, you can see this dominance of sockeye salmon in 2022. And it was interesting that in both 19 and 20, chum salmon were the #1 species, followed by coho. That's certainly very different. Now you might argue, well, these are different geographic areas. So next slide shows the 2022 catches just by the Franklin and the Shimada, which were, more or less line up with the earlier 19 and 20. And again, we still have a lot of sockeye and not very many coho compared to these previous years. Next. So one of the big questions we have for the survey is, what's regulating where these fish are? And one of the simple ones to look at is temperature. And this is just kind of a first cut at the data. It's showing each graph is for a different species. The x-axis is the temperature. The y is the fish per tow. So this is each ship. I just took the raw numbers that I had from each one. And there's a key over there. The greens are Tin Row, reds are Franklin, triangles are the Northwest Explorer, and the blues are the Shimada. And you can see that there's definitely some differences among the different species in what temperatures they're caught in. And the bottom right bar graph is just showing the mean temperature by species and ship. And so you can see that sockeye were on average caught in much colder waters than things like pink or coho salmon, and chum is kind of intermediate. And we know this. This is has been found in previous surveys, but it's kind of nice that we're getting the same results. Next. And then we also mentioned that, a lot of people have talked about interactions between sockeye and pink salmon that influence sockeye productivity, just because the pinks have such a strong even odd year signal. And it was really interesting that they really do not co-occur out over there, out in the North Pacific, especially in winter. So the graph on the left is showing the abundance of pink salmon in a hull, and on the y-axis is the abundance of sockeye salmon in the exact same hull. So other than one dot out in the middle, you can see that most of the points line up either along the pink salmon axis, which means that there's lots of pinks and no sockeye, or along the sockeye axis, so lots of sockeye and no pinks. And the maps also just kind of illustrate that the areas that we got a lot of sockeye, we didn't get a lot of pink salmon. Next. Okay, Dick.
So I'm going to talk about, we made what I consider a number, I think, is major discoveries in 2019, 2020, and 2022. And I'm going to talk about just about the schooling of, in this case, coho, and a little bit about chum. Just a little bit of background. Russian scientists, Shuntov in particular, have been publishing for years that salmon do not school. So the discovery that salmon can form large schools, I think, is a fairly significant contribution. So this is coho, and this is a 2020 survey. And in this particular set, we caught 98 coho, and we came back to this same location about 2 weeks later, and we didn't catch any. And in fact, in the stations close to this area 2 weeks later, there really were very few coho at all. So clearly, the large aggregation of coho that we caught in this set number 5, had moved in some other area. And when we looked at the composition, the stock composition, this is the example of the particular populations. And you can see they range from southern US to Southeast Alaska. There's a Russian coho in there, and that may be a DNA issue. But the point still is, it is indisputable that this large school of coho contained a very diverse group of populations. And the only explanation is that as these particular populations left their coastal area, they were able to essentially find each other in the open ocean, and then we're going to propose remain as an aggregate or a school. And we suspect that this is for the winter. I'll show you another slide in a minute. Well, maybe I won't show you in a minute. I'll show you in a second then. And this is not part of the survey, either in 1920 or 22, but this was a survey that we did in the Strait of Georgia in 2010. And this was one large catch of coho, clearly a school. And again, if you look at the DNA composition, this is a number of stocks from different areas. So there is little doubt that coho can form schools. The schools are there in the winter. Our hypothesis is that this might be an adaptation to it. Normally, this is considered to be a way of minimizing predation effects. That's what the standard biological interpretation would be, which is a hypothesis. Our hypothesis is that the schooling is a more efficient way of feeding, so it minimizes the metabolic costs of feeding. And we'll see whether or not that turns out to be correct. Now, there is one other slide, I think the next one. And this is what I call Chrys Neville's historic slide. This is one gillnet panel, and there are seven ocean age 1 chum salmon in that little area of the gillnet. And the chum salmon have been identified from northern British Columbia, Southeast Alaska, and Central Alaska stocks. So there's no question, I don't think anybody would doubt that that was part of a school that got caught in the gillnet, but clearly indicating that young of the ocean age 1 chum in their first ocean winter, find themselves and can aggregate in a school. So we have some evidence for schools for sockeye, and I think that now that we know that schools do form, we can now look at the potential implications. And this is Laurie's Weitkamp's comment that there probably are some political implications if it turns out that schooling in the winter, at least, is as prominent as we think it could be. There was one other, I mentioned at the beginning that one of the reasons that Brian and I were interested in the winter surveys was the ability to forecast the relative abundance of returning fish, in this case, chum salmon. And as you saw in the slide that I show you, that I call the collapse of the British Columbia commercial salmon fishery, chum returns to British Columbia have been very poor. And if you look at 2020 and 2022, now the catches are small, but you can see that there really are relative very few ocean age 2 and ocean age 3 chum salmon from British Columbia. And I think anyhow, that this is an indication that these surveys will identify when there are going to be poor returns, and of both pink and chum. But this is, as I said, the sample size is small, but again, because it is a survey distributed throughout the survey area, and because we are seeing poor chum returns, I think there's a possibility that these surveys are a method of forecasting when there are going to be poor returns of chum salmon. And then the final slide that I have is, it's been mentioned about the steelhead catches. Now, this is extremely preliminary information, and it most definitely will change in terms of the numbers, the ages, and maybe not the length frequency. But once again, you can see from the length frequency distribution that there are at least two ocean age classes. And then on the graph or the table in the bottom are our preliminary estimates of the ages of the steelhead that were in our catch. Now, we have to go back and look at some scales again, and we want some people that are better or more familiar with aging steelhead than I was. But there's no question that that the steelhead were in the surface meters, 2 meters of the ocean. And our hypothesis is that steelhead essentially are genetically programmed to stay in the surface meters, 2 meters. And I think this, Chrys, I think there's probably maybe the bottom of the slide. I think there, oh yeah, there it is there. 7 of the 57 steelhead we got were clipped, which may be a smaller number of clipped steelhead than we at least expected. Now, we haven't got the DNA back. We have diets looked at, but there's a lot of work to do with these steelhead before we better understand exactly where they're from and exactly how they are surviving in the winter in the Gulf of Alaska. That's the end of my slides.
And now I'm up, excuse me. I'm going to be talking about sockeye, and I'm going to start talking about what we actually saw in 2019 and 2020. So as you can see from these two figures, the majority of the sockeye in 2019, of the 73 sockeye we caught, were caught in the northwest quadrant. And in 2020, we weren't able to fish that area due to weather, but the sockeye that we did encounter, which were 51, were still more northerly distributed. Next slide, please. What's important about this data is that the stock DNA showed that there was a really broad mixture of stocks caught in both years. In fact, the most westerly fish were identified from Kamchatka in Russia, and the most southerly from the Columbia River. And the Bristol Bay sockeye were probably the only ones that had some kind of regional component to them, with most of those being caught in the northwest quadrant in 2019. The rest of these fish were really well mixed. So now I'd like to go from looking at all of the stocks just to looking at our Canadian fish. And this next figure is the catch of only Canadian origin sockeye salmon. And you'll see the survey area in the Gulf of Alaska, and then you'll see a transect that goes out across the North Pacific. And in 2019, we were fortunate that the Russians fished this transect in February en route to Vancouver. And they took mussel samples that we were able to do DNA from. That most westerly point at 176.2 east is possibly the most westerly record of a Canadian origin sockeye salmon. And what's important about this is our Canadian fish were distributed over 4,000 kilometers across the Pacific. Also, what's important about this is most of those fish in the Central Pacific were first winter sockeye salmon. Now you'll see about 6 or 7 of these red circles actually have a green star in them. And that's to illustrate where we caught Chilko Lake sockeye salmon. So this is one stock of Fraser River sockeye that were distributed over that 4,000 kilometers from coastal BC right into the Central Pacific. So the two take-home messages from this past data is that these stocks are mixing well with stocks from other countries, and there is a broad distribution of our Canadian sockeye. So now let's go to 2022. These are the preliminary results from the 53 sockeye salmon that were caught in the raw spirit. And you can see that we have a mixture again. 36% of these fish were from BC and 28% from the Fraser River. We also caught a number from Alaska, a large number, and a couple from the Columbia River. The Columbia River ones actually included one that was from Okanagan Lake. And we all know that 2022 is an Adams River year, and the Adams year is typically dominated by late run sockeye. So we wanted to take a look at what we were seeing in the Fraser River fish specifically in our catch. And what we found, going back to the forecast, for the 2022 forecast, at present, they're forecasting about 38% late run and 45% summer run, which is already giving an indication that the late run is lower than historic numbers would have been. But in the raw spirit catch, we saw only 14% late run. We did fish only in the southern areas. So we combined this from data that Jackie King and Cam Freshwater, who were the chief scientists on the Sir John Franklin, provided. And even with their catch, in the Eastern Pacific, we are only seeing 15% late run sockeye, with the remainder being summer run. We don't know how to interpret this yet. It's early. We do not know if this is an indication of poor survival of the late run stocks, or are they possibly rearing outside of the study area where these two vessels fished. We don't have the DNA back from the other vessels yet. We hope to get that back. And obviously we're going to have the returns coming in soon to the Fraser River to help us understand if this is an indication of what we should be seeing coming back this fall. Next slide.
Okay, couple slides on predation. The reason we bring this up is that it's always been hypothesized that the primary source of mortality on the high seas is predation. That for whatever reason, fish get sick or, are skinny, they're starving, but they get eaten, that's how they get killed. Next. And the Russians have looked at this a lot based on scars and wounds on fish. And these are kind of what they've identified as the major or most likely predators on the high seas. So we have some fish on the left, lancet fish, daggertooth, and then two sharks, spiny dogfish and salmon sharks, and a variety of marine mammals, porpoises, dolphins, toothed whales, and pinnipeds. Next. And what's interesting is that we really didn't catch very many of these at all, or see them out there in the case of the marine mammals. It's not like there's tons and tons of predators out there that would explain the mortality that people assume happens on the high seas. Next. Now you might say, well, what about the ones you didn't see or you didn't catch? And that's where this environmental DNA comes in. So this is work from 2019 and 20 surveys. So the orange triangles are 2019, the blue triangles are 2020. These are where Christoph Dieg detected DNA from the different potential predators. And you can see that, yeah, obviously we missed some salmon sharks out there, but there's not lots and lots of predators out there. Next. Which suggests that if predation isn't a major source of mortality, then what is? And I don't have a good answer for that. So just really intriguing. Brian.
Thanks, Laurie. So Laurie leads to the obvious question is what's coming in the future? It's not going to be a surprise to anybody. We believe there is serious value in continuing some of this work. The UN Decade proposal that has been submitted and endorsed by the IOC would continue some of this work. And our reasons are maybe down to three big points here that I wanted to briefly touch on. One of the things that we're doing under Becky is working on workshops, and we've talked to a number of the global climate modelers and people that work locally in the season to decadal downscaling of global models to more localized models. And they make a really important point. Their point is that they're not bad at building projections over the 30, 40, 50 year trends, and that they really talk a lot about. The skill level, though, is very poor in projections over the short term. And there they talk about their skill being low. And the consensus opinion of these modelers is really that in the short term, and I just use this range of 5 to 20 years, we have to live with climate change and climate impacts as the world adapts to changes to reduce the overall impact eventually. But what we have now is what we have for the next couple of decades. And the advice is you simply need more informative and better data to inform decision makers and our communities. So being out there is really not something that we should do if we want to. It's actually probably a requirement for really making good advice for appropriate management. The other thing is that when we sit down and talk as a scientific community, and we did this in a virtual workshop after the 2019 and 2020 cruises, this is now documented in the NPAFC technical report number 18. But when we really talk about what we saw out there, we know very, very little about the interactions of the species. And you can see the photos, but Chrys and Laurie have been out there. When you fish at night with these trawls, we catch big abundances of euphausiids, myctophids, and squids. I think we've identified 19 species of squids now, obviously some being more abundant than others. But the bottom line is we know very, very little about the interaction between these ecosystem components, certainly not enough to build models around. And I think that one of the things that we all want to emphasize is that we really have built an international community. And if you're out there and you look at the areas sampled, that's a massive area, right? No single country in a particular survey is really going to provide sufficient information. So these big international collaborations really are a requirement. The situation with Russia is sad for many, many reasons, but for Pacific salmon, we probably have the reality now that maybe Russia's not engaged for the next decade or more. All right, so there really is an onus now to Japan, United States, and Canada to really get out there and do this job. I think the other part is that as we talk to companies, actually using the information and the technologies they're building, getting out there and doing it's going to stimulate more innovation and technology. We're going to be able to do more things better and more cost effectively. We're going to be training and providing new opportunities. And it's probably the only way for us to really inform our communities about climate impacts and what are our governments doing about managing the reality of the short term, because the effects are very obvious to everybody. The ability to forecast or predict these in the short term is not well understood, but the modelers tell us that it's not going to happen very quickly. So we really need to get out there and get the data. We'll go to the next one. Laurie, I think I'll just do this one to finish up. And that. So the PSC, of course, deals with this, right? I can talk openly now because I'm no longer a commissioner. And that, sorry, John. So the reality is, of course, PSC has to deal with all this variation and trend, right? But we also have a unique opportunity to really be strong leaders in the future. I mean, we are an international agreement. We have the voice of the governments with us. Being supportive of a big UN decade program like Becky would be a major way to proceeding. The management entities that we all represent and work in, they look after the habitats, or whether you're talking about freshwater, estuaries, coastal zones, and so on, and they do the core assessments. So we really can't study salmon in the ocean without the involvement of the entities, right? We've had similar discussions about mass marking like this. You need to have the entities because they're the people doing the work. It's no different in the high seas. We need support and collaboration. And as we do that work, we will identify targeted research that we really need to focus on. There's some incredible innovation being developed. Wildlife Computing in Redmond is coming up with a micro tag for salmon, much smaller salmon now that would be a satellite link. So we can actually measure the actual habitat in the ocean that small salmon utilize. The PBT baselines, we actually can't give you really high resolution stock identification because we haven't sustained the standardized baselines. And the situation in Russia is a major problem in this now. But the other side of that is that we could compensate for some of that by having very accurate baselines for places we can sample. The Salmon Commission obviously spends time talking about environmental indices. The ocean indices could give you more lead time and could be more informative, because as you get into the coastal zones, and this is really one of the major science questions, is the survival rate and the growth largely determined in the nearshore coastal zones as they go to sea? And then what happens in the open ocean? It could be survival is determined in the first year and the rest is about growth and maturity, right? We really need to sort that out. And in the workshops, that is a prevailing question for many people across the Pacific. And I think the reality is that if we keep this going.
So Laurie, why don't you start this?
Yeah, why don't we go to the next one? We just have 2 more slides, then we're done. And Brian can come back and talk a little bit more. If people have questions.
He was almost finished.
He was almost finished, yes. As far as what's next, as I mentioned, there's lots and lots of data, thousands of samples to process. So there's a huge amount coming. There's a IYS synthesis conference in early October in Vancouver, BC, that a lot of people are going to be coming and bringing their results of the physical oceanographers, biological oceanographers, et cetera. Many reports and papers. As Brian mentioned, this Becky project, this basin scale events to coastal impacts, they have a really nice website, this becky.info up and running. The science planning is underway, fundraising is underway. And then perhaps one of the big questions is why stop now? Dick is threatening to do another survey next winter. So we'll see if you can pull that off. And I think one of the really big benefits is that we have built this multinational salmon community around the high seas work. I have gotten to know, and I think all of us here on this call have gotten to know the Russians, the Japanese, each other really, really well in doing this work, having, I can't tell you how many meetings, preparing for it afterwards. And it's really made the whole thing much more rewarding and easy and constructive. So it's really been great. And with that, we will open it up for questions.
From John Lamont, we have the question, does hatchery release pinks or any salmon from Alaska, Canada, Washington, Japan, Russia, Korea, or other countries impact wild salmon species nutrients in the North Pacific? And what impact would that possibly be on these wild Pacific salmon?
I think that the answer is that we don't know yet, especially from 2022, because the otoliths haven't been processed yet. So most of those have, at least the Alaskan fish, and I don't think other areas, but they're all otolith marked, so you can tell which hatchery it came from. So that's one of the big challenges, is identifying which fish is hatchery versus wild. And I think once we get that information, then we can start looking at that and seeing, are they caught together, for example? What's going on? Does it look like there's interaction? So that is one of those many to be determined questions but we should be able to address it.
Yeah,
Chrys. Yeah, Scotty, go ahead.
No, I was just going to say, I think that's one of the really big questions that we've been disappointed we can't address because of the pinks. We do see some of chum, but even those catches are relatively small. In the workshops I was trying to refer to, I guess I was cut off. There's quite a bit of interest in how early in the coastal zone that these effects may occur. So some of these interactions may occur in the coastal, even sort of tidal continental shelf waters, as opposed to the open ocean and so we really don't have a good answer for that question yet. It's one of the big ones that's really to be addressed.
Okay, thanks. We'll go to another popular one from Jim Mervin. He says, thanks for the presentation. And comparing catches in IYS surveys with earlier high sea surveys, what were the primary differences in terms of species distribution and relative abundance of various species between the 2 sampling periods? And I assume what Jim's talking about here is the pre-2019 surveys.
We're still in the process of comparing the historic catches from the gillnet work that was done, if that's what you're talking about, Jim. So that's ongoing, and we'll be able to report on that over the next month.
So just as a follow-up to Skip McKennell has been tasked with going through a lot of the historic data and making sense of it. And I sent him the sockeye and chum length weight data, and he plotted it versus the historic data, which is like 25,000 fish, and they lie right on top of each other. So indistinguishable. So the condition of the fish looks like it's identical to what historically we were seeing. So, but there's a lot of digging still to be done.
Okay, but in terms of distributions, Laurie mentioned it a number of times, and it is a really substantial discovery. Now, this is just the winter, but very, very little overlap between sockeye and pink. And there's a number of papers that have identified the overlap as a as a competitive problem. But in 3 surveys now, we see very little overlap. We are seeing pink salmon distributed, let's say, towards the south. And as Laurie had a nice slide showing very little overlap. We also see distribution differences between day and night in terms of our catches. And I don't think that's in the literature. So if you see coho as an example, if coho are predominantly in the surface waters at night and feeding, probably at dusk, and then deeper during the day. So they could still be in the same area of the ocean, but they could be feeding at different times of the day. And if you remember, a person a long time ago discovered that animals can be in the same area but use it differently. And that was Mr. Darwin.
Okay, thanks. Taking another one from Jim Irvin, he asked, did you learn enough with the limited gillnet sampling in 2022 to recommend continuing this sampling approach in the future? And do you think more effort should be spent in the future to sample at night?
You want to start that, Chrys?
Yeah, I'll say that if we want to study steelhead, we should be using gill nets, obviously, because that's where we were catching the gear or catching that species.
Jim, I think one of the things that we've learned with the gill nets, and we did have a number of the Japanese scientists warning us that they were very concerned about the effectiveness of the gill nets in the winter. This year was very difficult. Laurie's temperature plots and that across the 3 years was really interesting because 2019 we had some beautiful weather out there and we didn't lose many days at all. 2020 was more mixed, but we still had pretty good weather. This year we lost at least a third of our days at sea, and Chrys sat out on the raw spirit in some 9 and 10 meter seas. I mean, it was a rough environment. And the gill nets are not able to go out. I think, Chrys, you decided anything over about 3 meters you shouldn't deploy.
Effectively, we could fish the gill nets to 2.5 meters, and if there was no wind on top of that, we could fish them to 3 meters. But after that, they weren't fishable.
But that's
One of the things that.
Sorry,
Dick, I was just
our
going
first attempt. I mean, you know, this was using a Japanese gill nets. I mean, there are other ways of doing this that we could try.
If I was to do another survey, I would have the trawl as the primary, and I would have the gill net on board to sample both when we were encountering fish and when the weather was fair. So I think there's utility and there's a use for having that gear. It also allows, like I said earlier, us to study steelhead.
But I think we have also had discussions that we really tried to focus on the winter ecology, and that's a very tough environment. And we may get more for our dollar, and it's difficult to put these groups out there. We have limited numbers of ships. Privately, none of our programs cost less than $1 million we had to raise. And that BC Schrift provided NPAFC with, I think it was $3.8 million. So these are big costs. And maybe we should be looking at the reality of going in mid to later spring. And that to make the most of our efforts on the high seas. And that, so.
Well, we definitely need to do the spring. Let's call it early spring. Right? And we need that information as we continue to do more of this. I just want to repeat one more time that the commercial salmon fishery in British Columbia has collapsed. That is indisputable. And unless we have an explanation for that, then we have to accept that there is a much bigger picture that we need to know. And my argument is we need to better understand what's happening in the coastal ocean and in the open ocean. that we need a much bigger picture of what affects salmon abundance. It makes no sense to me to continue doing what we've been doing when we can clearly see that the commercial salmon fishery in British Columbia has collapsed. I mean, there's either we've done astonishingly bad management over the last 50 years, or that's telling us that there's information that we need to really improve our overall stewardship.
We had a question from Scott McPherson, ADF&G, who talks about PBT. Whoops, sorry, and his question is, how will wild stocks be represented in any PBT analysis?
Well, I think the reality is it won't be. But in terms of distribution, you could still learn a lot by having PBT with the accuracy that you attain from that by using your hatchery stocks to look at distribution. There are questions that will not be addressed by PBT, but then we can go back to stock ID and that with microsats and such. I mean, we have a better information basis with microsats across the North Pacific. Even there's some differences with Japan and Russia. But PBT is not going to give us many really big insights on the wild stock.
And it's Beamish again, and I try not to interrupt too much, but remember that we're going back to British Columbia, all right? And our commercial fisheries are largely pink and chum. So this is where we want to better understand what's affecting their abundances.
So Dean Allen from DFO asked, you mentioned you did not expect to catch many Chinook. Is this because they don't migrate as far north as the other species?
No, that's not quite right. Chinook are more complicated because there are different races of Chinook. One Chinook is not equivalent to the other, or sort of thing. So we have spring, summer, and fall. They have different migration patterns. They have different distributions. Fall Chinook, we very, very seldom see on the high seas. We see them in the coastal zone. They are highly migratory. They do go north. We've sampled some in the Bering Sea. Summer Chinook are in between. Spring Chinook are a different beast, and we do see some evidence of spring Chinook in the high seas. And that's consistent with some of the old gillnet sampling as well. But the rarity of Chinook out there is going to make really sorting this out a little, is going to be fairly slow. And that maybe we could do something with the new tagging technologies and that. But I think that the difference here in Chinook is that one Chinook is not equivalent to another. All right. And so we have to be careful how we describe them.
Chinook tend to be deeper in the ocean than other species of salmon, and so they're probably below the nets a lot. But we did catch a few.
So I've got a question. In one of the moments when Dick was talking, he mentioned the political implications across nations of coho schooling and mixed stock aggregations. Can you enlighten me there, Dick? What are the political implications that you see?
Laurie's going to answer that.
I think I will. So I think it's really, it reflects that this is a shared problem. Our fish are not separated from each other. Our fish are all mixed together. I think, Chrys's data on the sockeye, where these fish are coming from, caught together, they're all mixed together. It's not like we can, the US can go out and say, this part of the ocean is mine, this is where my fish are. That's not how it works at all. They're all mixed together. And so it behooves us, especially, be able to do, one nation cannot do this large of a survey. We have to work together and recognize that our fish are working together or living together. And so it's a shared problem. So we have to cooperate to understand what the ocean ecology, winter ocean ecology of salmon, because it affects all of our populations. That's the political.
Just think about, how did those coho all find each other? I mean, it's indisputable that they were in a large aggregation, whether you want to define it as a school or not. And it's indisputable that it moved around. And now you have dozens, maybe hundreds of populations that came from right from Oregon up to Alaska that all found each other in the early winter, right, and stayed together as a school. That's a hypothesis, but it's probably a reasonable understanding. And we hadn't considered that before.
John, there's a similar consideration with the IUU issues in the North Pacific. I mean, the unreported and unregulated. I'm concerned that our gillnet catches could be implying a significant effect of IUU on steelhead. We've seen some very serious declines in recent years in steelhead. I mean, we've got obvious problems in the interior Fraser, for example, that's kind of long term. But if IUU is expanding at the level that we hear people talk about, I'm very concerned that it could be having a disproportionate impact on the least abundant salmon. All right, so for us to get 57 steelhead with the limited gillnet fishing that we've done in that area, there's a lot of fish out there. All right.
Okay. Eric Taylor here from the Secretariat asks, was there evidence of the different salmon species hitting different vertical sections of the gill nets that could hint at differences in catchability?
Well, we definitely, that's one of the things we tracked is we basically divided the gill net into 4 vertical quadrants. And we have record of where these fish were caught in each one. And yes, there is differences in where they were caught, including the steelhead, which were typically caught at the surface and even right in the float line. That's still being worked up, and we'll have to take a look at it. If you looked at that figure that Dick showed of the chum salmon, they were all at the same depth range. They were swimming, they were caught right in the middle of the net, so at the 50% mark. You know, our chum catches aren't large enough to say something about overall in terms of that vertical distribution, but that information is there.
Okay. One of our repeat customers, Jim Irvin, says Chinook are strong swimmers and may be able to avoid the trawl net. Do you agree?
I have video showing juvenile Chinook this big swimming out of a trawl. So yes, we know that they would get picked up with the eDNA, and that's the power of that. And we don't see them. We don't have the eDNA for 2022 yet, but in 19 and 20, it didn't look like there were lots of Chinook out there that we were missing based on the eDNA samples we had. So, but yes, I think all these salmon are probably good enough swimmers that they can swim out of these trawls. There's a pretty good pressure wave in front of them, and that's really one of the big questions: is what is the catchability of these trawls? That's why I think it's really important that we, use alternate gear, whether it's a gill net or long lining, other things to get a sense of what's out there. And I think especially the larger, sharks. I was amazed that we caught one. 2 got caught. Because they definitely can swim out of a net without too much problem.
And Chrys, you had 2 salmon sharks, right? That neither of them were able to be landed?
Neither of them were landed, but yes, we had 2 salmon sharks. And going back to that, if you remember in 2019, we had GoPro cameras in the trawl nets. And there was another example of schooling actually into the net as a school. And we saw a proportion of those fish actually leave the net. And in that sample, the catchability agreed with what the Russians were using, but I mean, that was just an example.
Chrys, that's an interesting point, is that the catchability based on that one trawl with the GoPro was what, about 0.385 or something like that?
30%.
Yeah, and very similar to what the Russians estimate the catchability is for their assessment surveys.
Yeah.
You have a question in the chat from Gary Morishima. He asks, are there any plans to compare bycatch from the trawl with winter survey results?
Yes. So we'd like to, I'm waiting for all the data from all the ships, but trying to look at non-salmonids in the catches. They're really, one of the objectives of the entire survey is really trying to construct the entire ecosystem. And we're using trophic biomarkers as one way to do it. And the other is actually just the catch and look at that and see, where are these different non-salmonids out there? Squid, mactophids, jellyfish are the three big groups that we saw a lot of. And their abundances dwarf, in many cases, the abundance of salmon out there. So yes, we are hoping to look at that. It just hasn't happened yet.
But
I think that's salmon aren't the only things out there. Go ahead, Dick.
There's very few other species during the daytime in the Gulf of Alaska in our surveys.
It's interesting though that what there is, both Jackie King survey and the Gillnet survey saw, similar other species caught, including black rockfish.
Yeah, but there's only half a dozen.
Dogfish. Oh yeah, there's not very many, but they are similar.
What is amazing is that there are very few species other than salmon in the surface waters of the Gulf of Alaska in the winter, in the daytime, including predators.
So there was also an exchange within the Q&A windows here, and I'll read out the question and a couple answers, throw it open to the panel if you want to augment this. Nick Brown asked if the individual stock IDs for the sockeye would be available in the coming months. He says it would be very interesting to see the extent to which different Canadian stock distributions overlap. Christopher Havoc from ADF&G said that ADF&G will have the results for the October PSC meeting. And Chrys, Neville responded, I'm not sure what stocks would belong to this group specifically. However, about 50% of our sockeye catch in the gillnet/longline that could be classified to a specific Alaskan river were from north of the Aleutians.
I think I answered the wrong question there. I was answering John's question about what percentage of the 60% Alaska fish.
I got it. Yeah, I couldn't see the matchup there.
I clicked on the wrong one. And so in that case, about 50% of our Alaska fish were from rivers north of the Aleutians, but I'm not sure exactly which stocks he was querying.
Yeah, that was a earlier question from John Lamont asking about what percentage of the Alaskan fish were bound for northern Aleutian, Yukon, Kuskokwim.
And regarding the Canadian sockeye, a lot of the information from the first 2 years, there's a Winter Ecology Conference publication from NPFAC. It was a conference that Dick and Brian organized. A lot of this information from the previous years is in there, and including the sockeye information. But that data is all available.
I am not. Let's see. I'm going to take a quick look at the questions we've got open. Jim is going for the gold here. Jim Urban asks one more question: are these fish really schooling, or is it just that each species has distinct prey and water temperature preferences, and they tend to congregate in areas of highly suitable habitat? In other words, they're not seeking others of the same species, but rather just in highly desirable species-specific habitat.
Well, didn't he just define schooling? I mean, Dick said it's a behavioral adaptation. I mean, as an adaptation, we're a long way from proving that, but I think the way Jim describes it is probably why much of them school. There's probably a predator avoidance element to this. But at the same time, I mean, I think the reason they would be congregated and how they find each other are such preferences. So, I mean, there's no question that the distribution of the fish, densities are relatively low until you hit one of these very heterogeneous aggregations. And we've had, for the amount of effort we had out there, we've seen a few outstanding examples. The one on the Russian trawler was 680 1 year sockeye and one trawl set. I mean, that's astronomical compared to anything else. But equally so, in 2020, in the southern margins, Chrys, what was the final number for the coho? 89 coho in 2 sets, right? And very little else, right? So, and one of the other really interesting things that's coming out of the workshops is that people that are working with the phytoplankton also are talking about significant heterogeneity in the biomass of the plankton. That's so, and I think it is a relationship to the habitat that they're using.
I think
I don't think there's any question that there's evidence that fish are schooling. And if you think about it in the opposite way, there's no evidence that they don't school either. But after being out there now for 3 expeditions, and as Brian, he described it as heterogeneous, patchy could be another way of describing it, with the odd time in which you'll get a larger catch of, let's say, 20 or 30 fish or larger. Very, very rare do you get anything like that. And if, I can't remember the percentages, but a higher percent of fishing where we don't get anything. So and then the fact that the 2020 set number 4 where we caught, I can't remember, 80 or 90 coho in one school. And then looking at the stock composition, that's indisputable.
The other thing is, if we look at the gillnet catch, our gillnets were 2.5 kilometers long, and we had temperature sensors running along the length of them. And the catches were spotty, so that there would be groups of fish that were caught more than the odd fish here and there. And there was very little difference in the ocean conditions within one set.
Those 7 chum salmon that we showed you in the gillnet from, a half a dozen different locations, well spaced out along the coast. Those guys all found each other and were caught in a small aggregation. What we're seeing is quite remarkable. We're all impressed that salmon can find their spawning streams. Now we're going to be impressed that species can find each other out in the open ocean, and at least in some cases stay together over the winter at least. And we're going to hypothesize that that's an adaptation to more efficient feeding. It's a hypothesis, but there's a little bit of literature that supports that, by the way.
I think Jim brings up a really interesting point, though, in that, especially with the trawls, that you're really sampling over a huge area, a single set. We were towing for an hour. Some cases we were towing for 2 hours. That's 5 nautical miles that we are covering in an hour-long set, 10 if you do a 2 hour, and our speeds were comparable to the other ships. So unless you really can't look at the fine scale structure of where these fish are, and that's where I think the gill nets are really valuable, super valuable to look at that. And I had hoped to get some cameras in the net, because during the daytime, the surface trawls, you can just use an off-the-shelf GoPro, put it in there, and get great footage of what is coming in. So you can look at the distribution of fish as they're entering the net or leaving the net, as the case may be. And I wanted to do that with the Shimada, and we were scrambling to put the cruise together. I didn't have time to do it, but I think that hopefully if future cruises, we can really try and do something like that, or, a Ditson or something to look at where are these fish entering the net as you're towing it for these long periods of time. So it's important. And I think it really gets to your point, Jim, what is the really fine scale, ecology going on with these fish?
Okay, but Chrys already has done that. And Chrys, could you just tell them one more time?
About well, we did that for about half of the nets in 2019, and it was in all the daytime sets in 2020. Unfortunately, GoPros, they work amazingly, especially out there because the water's so clear. But we can't use them at night, obviously. So there is information for those 2 years there, and we did see those coho salmon. We have some other examples as well. I just don't think we've identified them quite as deep, or the footage wasn't quite as clear, so it's not.
But
You know,
they, for the tow that you have documented,
10
Where was
coho
it?
came in together, and
for how long were they in the net? It was just a short period of time.
they were in the mid part of the net for a short period of time. They came in together as a group, and then we could see seven of them leave, and we retained 3 in our catch.
So that answers Jim's question with respect to that one particular school.
We've used nets, cameras in the nets a lot on the Washington to Oregon coast. We're using a slightly smaller trawl, but the biggest challenge with that is that you got to get somebody to look at all the footage and count fish and identify them. And even summer interns don't want to do that. It's pretty, but it's really helpful. And I think that in future cruises, we should think about seriously trying to put more nets or more cameras in nets to look at this behavior and see.
The other thing that the cameras provide is we could clearly see the sea lice in 2019 on the backs of the sockeye, the chum, the coho salmon. So we were seeing what we thought were sea lice marks on the fish, and the video footage clearly showed that there was large abundances of Lepsalmonus, especially on the sockeye in 2019.
So Gary, Laurie, others involved in hosting here, we're at about the hour-and-a-half mark here at 3:30. I see some of our attendance is starting to drop off. Understand there might be a couple more questions that come in. I'll keep an eye out for that, but I did want to mention the next steps in the seminar series. We'll be taking a pause for August. There will be no seminar in August. In September, we are looking at the 4th week in September, so relatively late in the month, to look at freshwater habitat and productivity. We've identified Chrys Jordan from the Northwest Fisheries Science Center in NIMS to present. And during today's seminar, I got confirmation that Dan Selby from DFO will also commit. There's some mismatch in availability of dates, so we'll see if we can line up there in late September. And of course, as always, the Secretariat will be sending out announcements ahead of time, weeks and then days and then hours before the seminar starts. With that, I can turn it back over to Laurie and Gary if you want to have any closing remarks.
Thank you for coming. This has been great. And I should say we're going to have seminars, as John mentioned. We'll have freshwater habitat in September. We're looking at life history variation. Correct me if I'm wrong, Marisa, in October, and then hatchery wild interactions in November. And then we're hoping to do live seminars at the postseason and the annual meeting. So January and February, we hope to actually have speakers in person. So that'll be really a nice break. Marisa, do you want to say something about the October speakers?
Just that the October speakers from the US side will be Jan Olberger, and from the Canadian side, Brendan Connors, and they'll be discussing differences in salmon productivity related to environmental change and life history attributes. So that should be a great talk.
And that is October 26th, is that right, Marisa? Okay. So we can get people to mark their calendars now. And with that, let me check one more time. I think that kind of wraps up the question section. And I think we can wish everyone a happy Wednesday afternoon.
And thank you to our speakers today. That was great.
Indeed. Thanks to everybody.